Hello everyone, my name is Ojaswini Methania and today I'm going to be talking about the structure, function and analytical techniques of lipids. Starting with what are lipids? Now lipids are a diverse group of biomolecules that are involved in a lot of life processes from energy storage to cell signaling. Now lipids are water insoluble molecules that show solubility in organic or non-polar solvents such as ether, benzene, and toluene. This solubility behavior of lipids is a distinguishing feature that separates it from that distinguishes it from other biomolecules such as proteins and carbohydrates. Now, proteins show a lot of diversity in their function as well as their structure. To look at the structure diversity of lipids, these are some of the structures that a lipid forms. Now, the leftmost group, triacylglycerides or the triglycerides. The triglycerides are the simplest lipids that can be constructed from fatty acids. It is composed of three fatty acids that are each in an ester linkage with a single glycerol molecule. Now, if you'll notice here, all of these lipid components either have a glycerol or a sphingosine as a backbone, along with one or more long chain alkyl groups of fatty acids, which is shown in yellow, and the polar head group, which is shown in blue. Key lipid components integrate into membranes, shaping structure, communication, and stability across human systems. Now, let us take a look at one of the most structurally important form of lipid known as phospholipids. Phospholipids are central to membrane architecture. They show an amphipathic nature, that is, they are composed of a hydrophilic head and a hydrophobic tail, which drives them to self-assemble into bilayers, creating semi-permeable membrane essential for cellular integrity. That is, it forms the cell membrane, as you can see here. Now, these phospholipids serve as the fundamental scaffold of cell membranes, and thus, phospholipids lay the groundwork for membrane bound functions across the body. Now, let us take a look at some of the important biological roles that lipid plays in our body. In the brain, lipids are essential for the formation of the myelin sheath, which is a protective layer around neurons that speeds up the electrical signals. It acts as an electrical insulator that speeds up the electrical signals. In the muscle cells rely on the lipids as a dense energy source during prolonged activity. On the body surface, fat is stored in adipose tissues, which serves as a thermal insulator in subcutaneous tissues and around the certain organs. It cushions the organs and protects them from mechanical shock. In the liver, the liver is the hub of lipid metabolism. It synthesizes, modifies, and exports the lipids. It converts cholesterol into bile acids and steroid hormones and regulate lipid levels in the blood through lipoprotein production. Now let us take a look at the lipid metabolism in the digestive system. The dietary lipids are mainly digested in the small intestine by the action of the bile salts and the pancreatic lipase enzyme. Bile salts emulsify the fat, increasing the surface area for the enzymatic breakdown, and pancreatic lipase breaks down the triglycerides to form monoglycerides, fatty acids, and glycerol. Now, these individual constituents then enter the intestinal epithelial cells called as the enterocytes, where they are recombined to form triglycerides. Now, these triglycerides are packaged with cholesterol to form lipoprotein molecules, which are known as chylomicrons. Now, chylomicrons are a type of lipoproteins that enable transport of water insoluble fats within the aqueous environment. And the chylomicron molecule is shown here in blue. Now, these chylomicron molecules eventually leave the enterocyte cells to enter the lymphatic system and eventually flow into the bloodstream. The wall of the capillaries is contained a surface enzyme called lipoprotein lipase, which hydrolyzes the triglycerides to form fatty acid and glycerol, which then enables them to pass easily through the capillary wall into the tissues, which is after which they are either further oxidized for energy, or they can be re-esterized for storage. To take a look at the To take a look at this lipid metabolism on a biochemical level, we find out that the lipid metabolism is closely related to the carbohydrate metabolism. As we just saw, the triglycerides are broken down into glycerols or fatty acids. This process is known as lipolysis. Now, the glycerol formed upon lipolysis is converted into a glycolysis intermediate, whereas the fatty acids undergo beta oxidation to form acetyl coenzyme A, which further undergoes Krebs cycle to generate ATP. Now, the fatty acids are composed of 16 or more carbon atoms, and for each cycle of beta oxidation that it undergoes, two carbon atoms are removed from the fatty acids to generate one molecule of acetyl coenzyme A, which then generates ATP. Hence, we can conclude that fats produce more energy per unit mass than carbohydrates. Now, excess acetyl coenzyme A produced in our body gets converted into ketone bodies. Now, these ketone bodies are important, especially in the events of a glucose starvation, where these ketone bodies are an important source of fuel, especially for our brain. On the other hand, in the case of diets rich in carbohydrates, when excess acetyl coenzyme A is produced, it gets converted into fatty acids. This reaction is stimulated by citrate and is inhibited by excess of fatty acids. Now, fatty acids can be converted to triglycerides for storage or synthesis of other lipids by combining with glycerol, uh, which is derived from the glycolysis intermediate. This process is known as lipogenesis. Now let us take a look at the application of lipids. Lipid research impacts multiple fields beyond biology, such as medicine, nutrition, and drug development. Let us take a look at some of these. Now, omega-3 fatty acids are essential polyunsaturated fats that support our heart and brain function by reducing the amount of triglycerides, by lowering our blood pressure, and by preventing arrhythmias. In the pharmaceutical industries, liposomal delivery system use lipid bilayers to enhance targeted drug delivery and absorption, especially in the cases of anti-cancer and antifungal drugs, where they uh, where they improve tissue penetration and reduce the systemic toxicity. In the cosmetics industry, lipids like waxes and fatty acids hydrate skin, repair barriers, and improve product absorption. Lipid-based nanocarriers enhance the delivery of active ingredients like retinol and antioxidants deep into our skin. In the fuel industry, lipids from plant and animal sources are converted to biodiesel, offering a sustainable fuel alternatives. Biodiesel is produced by a process called as trans-esterification, and the resulting biodiesel is biodegradable, renewable source of energy, which is an alternative to fossil fuels with lower carbon emissions. Gene delivery. Lipid nanoparticles transport mRNA and DNA into cells. This is vital for vaccines and gene therapy. But not the least, let us take a look at some of the techniques used for lipid purification and analysis. Starting with purification. Here are some of the most widely used techniques for lipid purification, that is the false method, the Bly and Dryer method, and the Sockler reduction. Now, the false method is one of the most widely used methods for lipid purification. Firstly, the biological sample containing lipids is homogenized in a 2 is to 1 volume by volume ratio of chloroform and methanol. It is then proceeded by the addition of water or saline to induce phase separation. The lipids partition into the organic chloroform layer, whereas polar molecules that is metabolized such as salts and amino acids remain in the upper phase. These lower phases collected for lipid analysis and contains triglycerides, phospholipids, cholesterol, and other types of lipids. It is a relatively simpler method, and it also has broad coverage of lipid types and a very high efficiency rate. Now let us take a look at some of the analysis techniques for lipids. Lipids can be analyzed using using the following methods, that is thin layer chromatography, gas chromatography, or LCMS, that is liquid chromatography mass spectrometry method. Now, the liquid chromatography mass spectrometry method is regarded as gold standard for lipid profiling, as this method combines chromatographic separation with mass-based detection to identify and quantify lipid species. In this method, the sample is first injected into a column packed with a stationary phase and carried through by a liquid called the mobile phase. The sample components interact with the stationary phase differently, causing them to separate as they travel through the column. Different compounds in your sample will have different affinities for the stationary phase, so they will move at different speeds. This leads to separation of the components as they exit the column. The outcome you get is individual peaks on your chromatogram, with each peak representing a separate compound in the mixture. Once the separated compounds exit the LC column, they enter the mass spectrometer. Here, they are ionized, meaning they are converted into charged particles called ions. These ions are then accelerated through an electric field and passed into a mass analyzer. The mass analyzer sorts the ions based on their mass to charge ratio. In simpler terms, mass spectrometry tells us how much of each ion is present and the mass of each ion. Thus, this helps us to identify the individual compounds present in a sample with incredible precision. Lastly, I would like to conclude by saying as lipidomics continues to evolve, it empowers us to decode the lipid landscape with unprecedented clarity, transforming our understanding of health, disease, and therapeutic innovation. Thank you.